Okay, hello everyone. I'm Patricia Iweka from Hokkaido University. Okay, so today I will be talking about the non-destructive determination of individual cow milk quality and ovulation status during milking using online real-time near-infrared spectroscopy. Dairy farming is labor intensive. This is because it involves routines such as feeding, milking, manure treatment, and livestock management. Large-scale dairy farmers manage their livestock in groups, and this is a system known as herd management. However, for the production of high-quality milk, individual cow management is the best method. This is because monitoring the information of each cow is necessary for the production of high quality milk. And so there has been a strong need by dairy farmers for a novel technology that can help them to monitor the information of each cow during milking. I'm going to talk a little bit about progesterone because this has to do with the ovulation status. Now, progesterone is an hormone which helps to stimulate and regulate pregnancy, conception, and estrous cycle. Now, what's estrous cycle? Estrous cycle is simply the period of one heat phase to another heat phase. Now, looking at this chart, this diagram, you could see that as soon as the progesterone level, the progesterone concentration reduces, you can see this line here. It means heat. It means the cow is on heat. It means the cow is ovulating. And when the progesterone level increases and remains constant, it means that the cow is pregnant. However, when the progesterone level goes down again to a certain level, it means the cow is not pregnant, and it means the cow is ovulating. Now, it's the days of ovulation average 21 days. Okay. NIR spectroscopy has been applied in various fields of study. This include NIR has been used for the quality and the quantitative determination of agricultural commodities such as rice, wheat, fruit, milk, vegetables, and so on. NIR has been used for the determination of pregnancy in sheep. NIR has been used sometimes in 2001 for the determination of estrous cycle, ovulation in cow, but it was not successful. Now, the current method for the determination of progesterone concentration in cow, which will help farmers to know when the cow is ovulating or when the cow is on heat, has limitations. One of the limitations is that the method is destructive in nature. It has a lot to do with chemical. It consumes a lot of time, and it's usually done offline. And so there is no success yet for online real-time determination of cow milk quality and progesterone concentration of individual cow during milking. Over the past 30 decades, NIR has proven to be one of the most effective efficient method for the quality determination of agricultural commodities. This is because NIR spectroscopy is a non-destructive analytical method. It is quick, it saves time, and it is chemical and pretreatment free. Now, in our study, like I said earlier, we determine progesterone concentration. So the aim of our study is to use the NIR spectroscopic sensing system that we have developed to determine the milk quality and the progesterone concentration of individual cow during milking. Now I'm going to talk about the NIR that we used in our study. Here is the specification of the NIR spectroscopic sensing instrument that we used. It consists of three major devices, which is the spectrum sensor, the spectrometer, and of course the data processing, the kind of the computer that we use in obtaining the data that we used in our study. Now, looking at the spectrum sensor, it consisted of three halogen lamps, which is the light source. It consists of the fiber and others. The spectrometer consists of the optical density, the wavelength range, the photocell, the thermocontroller. And then the data processing computer that we use is actually a laptop computer, a Windows 7 laptop computer. And the spectral data were obtained in every 20 seconds during the milking process. Here is the overview of the NIR spectroscopic sensing system used in this study. Here is the line tube, the line tubes, the milk flow meter, the laptop computer, the spectrometer, the samplers, which is the plastic cups, the three halogen lamps, which is halogen lamp A, B, and C, and in here is the spectrum sensor. Now, this is the schematic diagram of the optical system. This is actually the spectrum sensor. This is what it looks like. Here is the halogen lamp A, B, and C. Here is the optical fiber and the glass tube. This is the original diagram of the spectrum sensor used in this study. So as you can see, here is the halogen lamp A, B, and C, and here is the optical fiber, and here is the glass tube where the milk enters, as you can see from here, and releases out from this end. So this is the original diagram. I'm going to talk about the flow chart, how the milking process was carried out. Now, here is at first, the milk from the teat cup cluster flows continuously from the teat cup cluster connected to the cow's teat, that's the cow nipple, flows continuously through a bypass, this bypass, into the milk chamber of the NIR spectrum sensor. The volume of the milk sample in the milk chamber of the NIR spectrum sensor is approximately 30 ml. The NIR spectrum sensor obtains an absorbance spectra through the milk and were recorded between the wavelength range of 700 to 1050 nanometer. And then the excess milk flow past the milk flow meter and then were released into the bucket, which we refer to as the bucket milk. Here is the milking mechanism. The milk from here, the teat cup cluster around this area, flows continuously through this bypass into the milk chamber of the NIR spectrum sensor. It obtains an absorbance spectra through the milk and then releases it into the milk sampler, which is into the plastic cups. Here is the milk flow meter, and here is where the bucket milk is, where the excess milk is being released into. 8 Holstein cows belonging to Hokkaido University dairy barn was used in this study. And the milkings were performed in 2 consecutive milkings, that is milking in the evening and the following morning for about 2 weeks, 2 or 3 weeks. Okay, so, and then the cows were used during their different lactation periods. In this study, we consider 3 major milk constituent contents, which is the milk fat, the milk protein, and the milk lactose. We also consider the solid not fat and the moisture content, which has a lot to do with the milk quality. Now, we also consider the somatic cell count, the number of the somatic cell count in the milk, which has a lot to do with the health of the cow. And then the progesterone concentration, which has to do with the estrus status, is the estrus status indicator. Now, for the reference analysis, mucoscan instrument was used to determine the 3 major milk constituent contents, which I said is the fat, protein, and lactose. And also the mucoscan instrument was used to determine the solid not fat, the moisture content, and the MUN. And then for somatic instrument was used to determine the somatic cell count of the non-homogenized milk. So in our study, we use the ELISA. We collected the milk samples for the determination of progesterone content. We collected the milk samples and put them in the refrigerator. at the temperature of minus 80 degrees Celsius, just for the chemical analysis. So for the chemical analysis, competitive enzyme immunosorbent assay was used for the determination. So we used the data we obtained as the standard data for the chemometric analysis. And then for the chemometric analysis, all the sample data set obtained in this study were randomly divided into two sample data sets. That is the calibration subset and the validation subset. Now, for the 2/3 of the samples, of all the samples were used to develop calibration models using the partial least square statistical method. And then the calibration models developed were validated using the remaining 1/3 of all the samples, which is the validation subset. So that is the remaining 1/3 of the samples was what we used to validate the calibration models that we've developed in this study. Okay, so in this study, these are the statistical parameters that we use to judge how accurate our results are. We use the R-square value, which is the coefficient of determination, the bias, the SCP, and the RPD value. However, we laid more emphasis on the standard error of prediction because this helps us to know how accurate our results are. Okay, so the lower the SCP values, the better the result for us. Here is the original spectral data, original spectra of the raw milk obtained in this study. So here are 3 peaks, 1, 2, and 3. The first 2 peaks was located around 740 and 840 nanometer. And this indicates the second overtone absorption of the carbon-carbon strings and the carbon hydrogen strings, which has a lot to do with the information of the fat content and other milk quality indicator considered in our study. And here is the strongest peak located at the wavelength of 960 nanometer, which shows the strong absorption of hydroxyl group in the water molecule, which of course the milk. Okay, so here is the validation statistics results that we obtained in our study for each of the milk quality indicator considered in this study. So here is the R-square value. As you can see, the fat content has the highest, an excellent result, which is 0.99. The closer the result had to 1, the better. The protein content as well, the result for the lactose, SNF, moisture content, and the somatic cell count, and of course the progesterone content. The SCP values were low. And so sufficient levels of precision and accuracy was obtained for the determination of each of the milk quality indicators considered in this study using the NIR spectroscopic sensing system. Okay, so here is the figure. Here is the figure showing the relationship between the reference fat content and the NIR predicted fat content. So as you can see, the RPD value was 0.81 and the SCP value was 0.17, which is a very good result. And this means that this result obtained can actually be used for the monitoring of the milk constituent content of individual cow during milking. Yeah, of course, the R-square value was excellent. Now I'm going to talk about somatic cell count. Somatic cell count is actually a universal indicator for milk quality indicator and the diagnosis, the diagnosis of mastitis disease in cow. Now, for example, when the somatic cell count produced in a cow is less than 100,000 SCC per mil, in the milk of a cow is less than 100,000 SCC per mil, it means that this cow is healthy. However, when it's above 200,000 SCC per mil, it means that this cow will probably have subclinical mastitis disease. So this says a lot about the importance of monitoring the SCC of individual cow during milking for individual cow management. So here is the SCC result. As you can see, the R-square value was 0.45, which is good. And this means that this result can be used for the diagnosis of subclinical mastitis disease because the precision and accuracy for predicting the SCC was good. Here is that of the progesterone concentration. As you can see, the R-square value was 0.72, which is a very good result. And it also means that this calibration model can be used for the monitoring of estrus status of individual cow during milking. So here is one of the studies of the progesterone concentration in our study. This is the result obtained for cow 1221. As you can see, the cow ovulated twice during our experiments. during this experiment. So the first ovulation date was 24th of July, sorry. As you can see, the blue line is for the chemical analysis result and the red line is for the NIR predicted result. As you can see that there is a similarity. The progesterone is down here, indicating that the cow is on it. And the second one was here, August 15th, which shows a very close correlation between the chemical analysis, and that's the standard analysis, and the NIR predicted analysis. So for the first time, NIR spectroscopic sensing system could predict progesterone concentration of individual cow during milking. And so estrus cycle, and of course, to know whether the cow is pregnant or not, using the progesterone concentration data obtained could be used. What I'm trying to say in essence is that the NIR sensing system obtained in this study could be used to monitor the estrus cycle and the early pregnancy of individual cow during milking. So the NIR sensing system developed could be used for online real-time determination of the three major milk constituent content, the SNF, the moisture content, somatic cell count, and progesterone concentration of individual cow during milking with sufficient precision and accuracy. Our NIR sensing system can help to meet the requirements of dairy farmers and veterinarians. It can help to give rapid feedback control for upgrading to dairy farmers for upgrading individual cow management. It can relieve dairy farmers of poor milk production and the poor milk production and economic losses. And of course, dairy farming can be achieved. Okay, so here ends my presentation. Thank you for listening.